Today, I'm pleased to introduce you to our speakers as follows. So we will first have Anne Gourmelon, who's the principal administrator of the OECD Test Guidelines program. She has a master's degree in environmental science and environmental toxicology from the University of Wageningen in the Netherlands. She joined the OECD Environment Directorate in 2002 after some years at FAO in the pesticide management program. At OECD, her goal is to maintain the collection of test guidelines relevant to the regulators and to the evolving chemical safety regulation, up to date with modern science and techniques, robust to generate reliable results and in line with animal welfare consideration. We have also Dr. Elijah Petersen, who holds a PhD degree from the University of Michigan in environmental engineering and a postdoc from the University of Joensuu, Finland, and another from the University of Michigan. He then joined the US National Institute of Standards and Technology, the NIST, first as a postdoctoral fellow and since 2010 as a staff scientist. He works in the Cell System Science Group in the Biosystem and Biomaterials Division. His research currently focuses on the development of robust, reproducible toxicological test methods. Dr. Petersen is an associate editor for the Nanotechnology and Nano Impact, and on the editorial board of the journals Environmental Pollution, Nanomaterials, Frontiers in Toxicology, and Environmental Toxicology and Chemistry. We will have also Dr. Gregory Goss, who has a broad program aimed at water research for improved environmental health and sustainability. With more than 200 journal research articles, 1 book and 3 patents applications, he's a leader in the field of environmental science. He's a professor in biological science and a fellow at the Joint National Research Council of Canada, University of Alberta Nanotechnology Initiative. Dr. Goss works jointly with industry, governments, and academia to examine the environmental issues related to water science. His research includes the effect of various biotics and abiotic stressor pollutants on aquatic animals. Micropollutants studied include nanomaterials, pharmaceuticals and personal care products, hydrocarbon contaminated fluids, and micro and nanoplastics. He also leads a large group focused on modeling future hydrology and water quality with future climate change. Finally, we have also with us Dr. Alan Kennedy, who has served as a research biologist toxicologist at the US Army Engineer Research and Development Center Environmental Laboratory, the ERDC, since 2006. He is a principal investigator on the persistent and emerging threats toxicology team in the effect processes and risk branch. Al has a bachelor degree in environmental biological from Michigan State and a master in science and environmental toxicology from Virginia Tech. His training is in novel methods for assessing the exposure and effects of environmental contaminants to biological receptors in water, sediments, and soil. He leads or co-leads military and civil projects investigating the implication of dredged materials, contaminated sediments, invasive species, metals, ammunition on range and advanced materials for use in defense application. I'll serve as technical lead of a ERDC on advanced and additive materials, environment, health, and safety research focus area, and he leads the ERDC's safe and rapid development of nanomaterials congressional interest. He's involved in efforts to standardize toxicology test methods with several organizations, including OECD. So with that, I hope you are happy to join us with this quite an impressive panel, and I will pass the floor to Anne Gourmelon so we can officially start this webinar. Thank you very much.
Thank you very much, Mar, for this introduction of our speakers today. I will just take a minute to check that interpreters can hear me.
Yes.
This is the case. That's good. Good afternoon, good morning, everyone. We are extremely happy that colleagues are interested in the subject matter of nanomaterial safety testing from various countries including countries in Latin America, could join this webinar today. It is the first time actually that we are providing simultaneous interpretation in English and Spanish in these webinars at the OECD on chemical testing and assessment. So I hope that you will find this webinar interesting and that you will learn a few things about OECD and the project that, the document that is now an OECD document, a guidance document for the safety testing of manufactured nanomaterials in the aquatic environment. So I'll take just a couple of minutes to provide a brief introduction, a brief overview of the test guidelines program and the share of nanomaterials related projects covered by the OECD Test Guidelines program. And also I'll take just a couple of minutes to provide some context around our test guidelines related activities. So on this slide, you can see the various projects we have distributed across the various series of the test guidelines. Section 1 on physical chemical properties, Section 2 on effects on biotic systems, Section 3 on environmental fate and behavior, Section 4 on health effects, and Section 5 dealing with other test guidelines. So the project that will be presented to you that has been completed last year is the only project that we had on the series, on the Section 2 of the test guidelines related activities, effects on biotic system. So it is published as OECD Guidance Document 317 and dated 2020, as I said. So what do we do? What is our reason to be at the OECD? We are in the test guidelines program developing quality standards for the safe assessment of chemicals, including nanomaterials, of course. So the OECD guidelines for the testing of chemicals are a collection of internationally agreed testing methods that have been harmonized and that are used by government and industry and independent laboratories to produce data for the safety of chemicals. Our test guidelines at the OECD are covered by what we call the mutual acceptance of data system, which means that once chemicals, including nanomaterials, are tested in one country, one member country or adhering country to the mutual acceptance of data system, then the data is accepted everywhere. In addition to test guidelines, we develop in the program supporting documents for the adequate conduct of guideline studies. These are called guidance documents, like the one that will be presented to you today. These guidance documents provide more detailed explanations on selected aspects of testing, like the conduct of the study or the data interpretation, for instance. Guidance documents provide more flexibility to adapt the guidance, the instructions to specific test or tested chemicals. So here on this slide, I'm going to provide you with a very quick overview of the projects related to nanomaterial safety testing that we have in the area of physical chemical properties here, ecotox, like the one that will be presented today, and also environmental fate and behavior. So in these little bubbles, you have the titles of the ongoing projects, and associated with the bubble is in a little square, the lead country, that is involved with the development of the product, the guidance document or the test guideline. And then there are interrelationships, of course, between these projects. As I mentioned here, there are certainly, of course, interactions between projects in the area of physical chemical properties and environmental fate and behavior. The guidance document 317 for aquatic and sediment toxicity testing that will be presented by our speakers today. Just a few words on the rationale. Why was it developed at the OECD? Preceding this guidance document, there has been a review of a number of test guidelines in the series 200 on effects on biotic systems in the aquatic environment. And the reviews have consistently concluded that those test guidelines, although they are generally applicable to the testing of manufactured nanomaterials, we denote in these documents the absence of guidance on producing and sufficiently characterizing test dispersion of manufactured nanomaterials before and during the conduct of the test itself. Also, there is an absence of requirements for measuring and providing nano-specific information, including, for example, primary particle characteristics, which is very important. Also, agglomeration and settling behavior in the test medium and transformation. Also, the frequency of these measurements, which is really important to really understand what's going on during the conduct of the assay. They are used in quantifying exposure-response relationships and use of other particle-specific response metrics, such as surface area or particle number, are unaddressed in the individual test guidelines that are mentioned here. So today, I will now leave the floor to our speakers who are going to provide in-depth explanations about this guidance document 317. And you can, of course, find additional information on our website then also at the end of the webinar, we will present you with additional information for where to find the recording of our webinar today. So now I give the floor to our speakers. Thank you very much and enjoy the presentations. Thank you.
Hello, everyone, and welcome again to our presentation this morning on OECD Guidance Document number 317, Aquatic and Sediment Ecotoxicity Testing of Nanomaterials. Thank you, Mar, for that very nice introduction, and also for the description of how this document fits in with the other OECD efforts. Next slide, Al. So in this figure, I show what our aim is, and that is to be in quadrant A. Each of these data sets is an example of data from a different laboratory. What we want to have is harmonized results. That is, different laboratories are able to get a similar result when performing a different method. If there is too many outliers or too much variance, you start to get into some problems. The OECD test guidelines have been established to be harmonized, when tested for dissolved substances. The challenge that we addressed in this guidance document is how to help perform these methods in a standard, Al, where'd the presentation go? Perform the presentation in a consistent way when testing engineered nanomaterials or manufactured nanomaterials, as I'll say during this presentation. As I'll describe in the next several slides, there are many challenges with manufactured nanomaterials that you wouldn't see for dissolved substances. Go ahead, next slide. For example, there can be challenges such as artifacts in the nano ecotoxicology testing that can occur at different steps. This could occur potentially for when you get your initial materials. They could be of different sizes, or there could be impurities there you may not be aware of. There could be changes to particles during storage. There could be issues from the dispersion process where you try to make if the nanomaterials are in a solid powder form, you try to make them suspended in a stock suspension or your test suspension. You could potentially change them if you're not careful. There could be issues with performing some of the measurements of the endpoints themselves. And this could be an issue, especially for in vitro test methods. There is a summary of, or a much more in-depth description of this topic in the paper that is shown below. One thing to also keep in mind is that while there is an OECD guidance document 23 on difficult to test substances, this guidance document could be interpreted to suggest to include particulates so that you avoid so-called physical effects. But that would be potentially problematic for manufactured nanomaterials because you may be excluding them, and that would change how you're doing the testing. So the suggestions in guidance document 317 are really focused on how to take into account this difference. Now, I guess I'll also take a step back too. I should mention that, as I'll mention again in another slide or two, that this was really a team effort. There was many collaborators from different countries. While Greg, Al and I, helped guide a lot of the revisions process. We had meetings with many people, and I also want to highlight Steve Diamond, who was a really key player during the first several years of this project. He's since retired, which is why otherwise he would be presenting with us today. But he was definitely played a major role in this in the development of this guidance document. Next slide, Al, please. To give you some examples of how testing for manufactured nanomaterials can be different than testing other substances. This is a schematic showing some of the behaviors that can occur. There could be a coating on particles that could be a food source, or it could be toxic itself. The suspended particles could agglomerate with each other and settle out of suspension. Some particles may dissolve and release ions that themselves may be toxic. And as I said already, there may be interferences in some assays potentially. These are just some of the examples of the challenges that you may encounter or may not when testing manufactured nanomaterials. Next slide. As I was saying, this was definitely a team effort. We had 20 different colleagues from 8 different countries that participated at some point in the development of this guidance document, whether it was from the initial meeting or during the review process. So these are some of the institutes that they represent. And again, on behalf of Al, Greg, and I, we really want to thank everyone so much for all of their contributions along the way. Next slide. So there are several meetings that led to this guidance document. One of the precursors for the development of this guidance document happened in Berlin in 2013, where some of the ideas about even making this guidance document came forward and helped shape the SPSF that was written. The first meeting that explicitly focused on this guidance document was in February of 2014 in Vienna, Austria. Then there was a larger meeting that was focused exclusively on this guidance document in Washington, D.C. in July 2014. We had 23 experts from 7 different countries represented. About half a year later, we met in 2015 in Dessau, Germany at the German Environmental Agency, or UVA. And then there was the last meeting at the ProSafe meeting in Paris, France in November 2016. So you can see there was many meetings where we discussed some of these tricky topics that I'll describe in the subsequent slides. Next slide, please, Al. The revision process for this guidance document was thorough and lasted several years. With the help of our different colleagues, we submitted the first draft in September 2017. We then had several rounds of revisions and comments. Altogether, we had nearly 1,000 comments, which it took us some effort to really consider them and to make the changes that they were suggesting. And it really, really made the guidance document so much better by the end of this process than it was at the beginning. So again, thank you to everyone who contributed by carefully reviewing and giving us critical suggestions on the guidance document. Then in July 2020, it was finally published and is available at this website. Next slide. Now I'll describe some of the recommendations from this project. All of these topics could have been things that we spent a whole half day at a workshop discussing. They're often complex and a lot of nuances are present in the guidance document itself. So first, we found in this, I guess to take a step back from what's actually in the guidance document, first I'll talk about a paper that came out as a follow-up from the 2014 EPA meeting. And this was published in 2015. The citation is at the bottom of the page. So we evaluated the OECD aquatic toxicity test guidelines for use with nanomaterials, and we found that they were generally applicable, but then some amendments were needed. And the amendments were needed, and they were distinct from those covered in guidance document 23, because there's a manufactured nanomaterial specific artifacts and key considerations. For example, the dimensions of the test container could have important effects on the assay results, unlike for dissolved chemicals. In this manuscript, we highlighted numerous topics where we were able to reach some initial consensus at our first meeting, but then also many areas where we did not reach consensus. These were then refined through the revision process of this guidance document, and we were able to reach consensus on many of them. In addition to some people had conducted additional experiments that helped reveal insights that help with the guidance document. Next slide. So these are the different sections in the guidance document. In the introduction, scope, and background, we described why we needed to have this guidance document, carefully considered what topics we would cover and what topics we would not cover, and provide some general background on manufactured nanomaterial testing. Then in section 4, we went into the analytical and measurement techniques that are needed to characterize the initial test material, and also the test material after it's dispersed either in a stock suspension or in the test media. In the section 5, we discussed the test dispersion preparation or ways that you could potentially do that. This is a very complex topic with many nuances. To cover some of these nuances, we'll have some of the flow charts from the guidance document will be discussed in the next several slides. The reason this is challenging is because there is a broad range of types of manufactured nanomaterials, and dispersion strategies that may work with one manufactured nanomaterial may not work for others. The conduct of the test covers some specific issues related to what happens when it's being conducted. For example, how to handle the dosimetry, as I'll describe in the next slide, or 2 slides. There are also in this section specific information for for different test guidelines where there may be unique considerations that we wanted to cover. So we had tried to have overarching guidance that would cover all the guidance documents or most of them, and then also more detailed guidance for some of the different specific test methods or test guidelines. Lastly, we give recommendations for nanomaterial specific data analysis and reporting so that the people who are conducting these tests and reporting them to regulators have guidance about what details they should include and which ones they can also consider including. Next slide, please. So these are some of the key topics that were covered. And as I was saying earlier, each of these, we might have spent half a day discussing. There's many important nuances here. So again, we discussed the characterization of the as-produced test material and the test material in the stock suspension and dispersions. There, we discuss how to do monitoring of the exposure concentration and consistency during the experiment. If there isn't enough stability, you may need to consider doing water exchanges, time-weighted averages, or other changes. This is important because for many test guidelines, they require that you make every effort to keep the concentration within 20% of the initial concentration. If you are unable to do this, we have some suggestions for how you can proceed. As I had just said, there are many different test dispersion preparation approaches because some materials may be very easily suspended in water. Some of them may be more challenging and hard to disperse no matter what you do. To handle some of these materials that are less stable, we provided in the guidance document a hierarchy of modifications to consider, such as for the pH, ionic strength, and potentially the addition of natural organic matter, although there could be issues with toxicity mitigation if you add that, so we have some additional considerations if you want to use natural organic matter. It is important to note that in some geographic locations, it is possible to discuss potential strategies with the regulatory bodies, and other geographic locations, this is not possible. Because there's many nuances here, I would also like to open up the discussion to Alan Steebed in case either of you have some additional input you'd like to provide at this time on any of these key topics.
Not for me, Elijah. Thank you.
Not for me either, Elijah. You're doing well, thank you.
Okay, good. All right, next slide, please. So we also describe 6 potentially relevant control experiments that you may need to perform to see if there may be a biased result or an artifact in the assay, or if you want to understand the relative components of exposure and toxicity. This could be relevant, for example, if you're testing the toxicity of a metal nanoparticle that dissolves, and you're wanting to understand, is the toxicity come from the particles or from the ions? As I had already described, we additionally provided assay-specific modifications for a range of OECD test guidelines. And we incorporated a section on methods for sediment spiking, because this is also was part of this guidance document in addition to pelagic organism testing. The last key topic was related to the different exposure metrics. We suggested as the default exposure metric to use the mass concentration. This suggestion was given because it is the easiest to measure. There are standardized tests that could be available for this, and it is the most ready for use at this time. In the future, however, it may be that other metrics will be better, such as the nanoparticle number concentration or the surface area concentration. If possible, and if the instrumentation is available, we suggest to try to also provide the data so that these additional exposure metrics could be provided. And with that, I'll open up with Al and Steve again to see if there's any other input they'd like to provide on this slide.
This is Al, not from Elijah.
All right, good.
This is Greg, not from me either, Elijah.
Okay, great. Well, with that, I'd like to pass it over to Al to dive into some of the additional content in the presentation with a focus on some of the different charts that were included.
Okay, I'm just needing to stop sharing my screen for a moment to change the interpretation. Just give me a moment. Okay, you should be seeing my full screen now. Can you confirm?
Yes.
Excellent. Well, thanks, Elijah. Thanks, everyone, for attending, and really appreciate the opportunity to present today. My name is Al Kennedy. I'm an ecotoxicologist at the U.S. Army ERDC. Just to clarify, our bios, we're presenting in a little bit different order than our bios are presented. In the next 7 slides, I'll provide a general overview of the flowchart logic that's included in the guidance document. And the text of the guidance document heavily supports how and when to use these flowcharts and indicates how the different flowcharts are connected. If you find these flowcharts too small in the slides in your screen, we invite you to follow along in the associated guidance document. If you don't already have the guidance document, I believe that the link was provided in slide 7. Also, for your reference, note that on each of these slides that the page number and the figure numbers are provided. So I don't plan to go over each box in the flowcharts explicitly, but if you have technical questions, those may be best addressed in both the Q&A box as well as in the verbal chat. So this first chart provides a general overview of how the guidance document flows. It's actually the first chart, and on the left-hand side, you can see the black boxes. That describes the process within the document in terms of how it flows and how to make decisions. For example, selecting the type of system you may need to test in, select important regionally or internationally important test species and methods. And it flows down the line to discussing dispersion methods for the nanomaterials, dispersion stability and how the nanomaterials or the manufactured nanomaterials may behave, how to spike and monitor the media throughout the bioassay, and then it goes into interpretation and input calculations. So the guidance document authors also recognized that there were several other OECD nano EHS related documents that were either in developed or published about the same time. So on the right-hand side of the figure, you can see the colored boxes where we either refer to this guidance document or other guidance documents on when to engage the techniques presented in those materials. So a very obvious connection is the test guideline on dispersion stability that can be used in concert to supplement selection of dispersion methods and measurements of the stability of the nanomaterials. And as Elijah mentioned, there's reference to determining how the animals are actually being exposed. Are they being exposed to both a particulate and a dissolved fraction? So it's very logical to reference the guidance document on agglomeration and dissolution that's in development. So along with this flow chart, there is a table listing the toxicity testing OECD TGs that we reviewed. And for the most part, the guidance document can be directly followed to execute those test guidelines. But toward the end of this guidance document, there is a section that has short paragraphs describing modifications that may need to be made for unique guidance documents, such as the algae toxicity tests, where there could be various interferences and controls that need to be run. So in this next flow chart, the logic process for initially attempting to suspend the manufactured nanomaterials with unknown dispersion stability characteristics can be executed in both an ultra pure water stock as well as test media. And again, these are pre-tests for when very little is known about the particular nanomaterial and how it may behave in the test system. These pre-tests are quick and easy experiments that can be conducted prior to the bioassay that inform the conduct and planning of the bioassay before use of any animals. Basically, just to make the conduct of the assay more efficient, to reduce the use of animals, and to try to get it right the first time. There is an initial dispersibility consideration in this flowchart and the associated text. If a stock can be made, there's considerations about whether a stock can be made or if direct addition to the test media is necessary for unstable nanomaterials. Stability kinetics of the nanomaterials, or basically how long they will stay stable in the water column during the bioassay, is assessed, and guidance to help select appropriate test methods and organisms for the water column toxicity test, as well as the sediment toxicity test, when to engage those particular test guidelines based on the dispersion stability behavior of the nanomaterials. There's also considerations on whether stability can be enhanced during the bioassay through slight modifications of water parameters, such as pH and ionic strength, or addition of energy into the system, slight shaking, for example. The usefulness of testing whole particles, the suspended fraction of particles, or the dissolved fraction of the particles only is also discussed if you need to determine the exposure vectors the organisms are receiving to see which component is necessary. Those are certainly options within the test guidance, but not requirements. So the overarching considerations that the subsequent flowcharts, in the subsequent flowcharts, cover each of these complex topics in greater detail. So the intent of this flowchart, which is figure 3 in the guidance document, is to determine if the manufactured nanomaterial can be sufficiently dispersed to make a stock suspension. And that could also often be in something like ultra pure water, milky water, low ionic strength water. If the nanomaterial is not dispersible, the flowchart and associated text provides guidance on how to spike the test material into media consistently so that you have similar amounts of nanomaterial across your experimental replicates. There are several different dispersion and media spiking options presented in the guidance document text. This is fairly difficult to visualize just from the flowchart and from me talking about it. So in the next slide, a cartoon is provided that illustrates the media spiking methods and serial dilution process to achieve a consistent nanomaterial concentration series. It's presented in the next slide. This flowchart also provides guidance on how to manage water exchanges if water exchanges are required by the specific testing guideline. And that's particularly applicable for chronic toxicity tests where water exchanges may be required or useful to maintain contaminant exposure concentrations or water quality parameters to support the test organisms. So this is the cartoon I referred to that shows media spiking and serial dilution methods. So I need to first provide the guidance document definition on manufactured nanomaterial stability. Within the document, this stability is defined as maintaining a consistent concentration within 20% during the duration of the exposure. And that is a guideline that is common to multiple testing guidelines, toxicity testing guidelines. And this also caused a lot of the debate whether that 20% should be applied for manufactured nanomaterials because they're dynamic and ultimately less stable in a lot of test solutions compared to more traditional contaminants that are assessed. And there was also a lot of discussion about agglomeration behavior, whether that need to be maintained, and things like dissolution kinetics. But often concentration is the most logistically feasible and cost-effective thing to monitor. So often the 20% consideration is in context of concentration, with a lot of considerations to monitoring and how the nanomaterials behave. So as mentioned in the prior flowchart, this illustration provides considerations for nanomaterial spiking and the serial dilution with supportive text in the guidance document. This really is based on the nanomaterial dispersion and stability characteristics that are specific to both the nanomaterial and the test media or test system. If a stable stock cannot be made, it's difficult to use that stock, so alternative considerations are used. So in option 1A, which is shown at the top, this may only be applicable to the most stable nanomaterials. It certainly would be applicable to things like dissolved metals. Unfortunately, nanomaterials don't behave quite as well as dissolved metals in terms of their stability, so it may not be applicable in most cases due to agglomeration and other behaviors that may cause inconsistencies in a typical serial dilution. So that brings us to option 1B, where you might have a manufactured nanomaterial that's stable in an ultra-pure stock media, but it might not be quite as stable or not stable at all in test media where you've got higher ionic strength. That might make a traditional serial dilution impossible, at least impossible in terms of getting consistent and replicable results in your bioassay. So you may consider things like individually spiking the test treatments or the test vessels, depending on how long you can keep the material stable. And it requires, this method requires robust analytical confirmation of the exposure concentrations, not only at the beginning of the assay, but also throughout. And option 2 considers when you have a nanomaterial that isn't very dispersible or at all dispersible or stable in the ultra-pure stock or test media. You may need to resort to individual spiking, either from a dry powder or a carrier stock medium with associated carrier controls. This is applicable to nanomaterials also where analytical confirmation is difficult and only nominal concentrations are possible in a typical lab. So this is also applicable to examples like carbon nanomaterials or carbon nanotubes that are in sediment media, where it's very difficult to well characterize the system in sediment. And it's particularly important to characterize the nanomaterials before they're spiked and to know your nominal concentrations pretty well going in. So of course, that's described more in the text of the guidance document. So the intent of this slide, which is figure 5 in the guidance document, this is more focused on, flowchart guidance on how to develop a plan to determine the manufactured nanomaterial stability in stocks that are often used in biological exposures. So this is creation of your stock media and how to determine stability. This includes decision guidance on how to quantify the exposure for unstable nanomaterials that result in a dynamic changing exposure throughout the duration of the bioassay. Unfortunately, there's no easy button here, and some expertise by the experimentalist is required. This consists of dispersion stability based on 20% of the initial concentration, and multiple sampling points and observations throughout the pretest may be necessary to make an assessment of the dispersion stability of the stock. So we now move on to flowchart or figure 6 in the guidance document. The intent here is to be more focused on assessing the dispersion stability of the nanomaterial in actual biological test media. This provides guidance on pretests informing the bioassay conduct. This is where there's more consideration, detailed consideration to pH and ionic strength effects that occur that may lead to instability of the nanomaterial in the test system. And again, the dispersion stability is based on maintaining 20% of the initial nanomaterial concentration in the biological exposure media. There are also considerations to whether consistent agglomerate size and dissolved metal concentrations can be achieved. That's more of a monitoring and characterization method, and also kinetic time point sampling may be required to adequately plan the bioassays in terms of water change frequencies, or at least monitoring frequency. And there's guidance on helping to determine if media manipulations are appropriate to increase the consistency of the biological exposure. And the logic for this and decisions are covered in greater detail in both the text and this flowchart. So finally, this flowchart provides more focused decision guidance on how to manage water changes and media renewals during the bioassays. Again, if that's required for chronic testing guidelines. There's consideration and logic for if and when it is acceptable to alter the pH or ionic strength of the media. So this includes relation to the isoelectric point by adjusting the pH or maybe the ionic strength away from the isoelectric point within species-specific tolerance ranges, which could be defined, for example, as 6.5 to 9 for pH. Throughout this process, the impacts of organism health are considered through reference toxicity tests. Altering the media may have implications in terms of the organism's sensitivity. And there's also description of the data usability of altering test media when the desire is to compare to historic data sets using the standard media. Any alterations to the media may potentially violate that. So that is also considered. So unless the media manipulations lead to an appreciable or significant improvement in the nanomaterial stability, the text in the flowchart guidance default to use of the standard test media with more rigorous monitoring of the nanomaterial state during the exposure at multiple time points. And that is to facilitate alternative endpoints or more robust endpoint calculations such as the time-weighted average to understand the dynamics of the nanomaterial exposure the organism is receiving, and also consider things like particle number and perhaps surface area, which would be a specific metric to the nanomaterial being considered. So that concludes the flowchart overview of this guidance document. Specific questions may be best addressed in the chat and verbally during QA, the question and answer session, since it's fairly difficult and time consuming to go through the individual boxes. But I'll now pass the torch over to Dr. Greg Goss to wrap up this presentation and discuss the path ahead for the guidance document.
Thank you very much, Al, and to Elijah and everybody for attending. We appreciate your attendance. As both Al and Elijah have alluded to, there was not necessarily a full compliant or full agreement on all the components that are necessary, as the complexity of working with nanomaterials, I think most people are aware, as certainly that additional layer into the testing guidelines. And one of the things that I think the guidance document tries to deal with is the variety of different characterizations that are needed. And the general recommendation is the more you can do to understand the nature of your particle and its behavior in solution, and this will help inform you of its potential toxicity and give you opportunities to look for proper interpretation and avoid misinterpretation. So the topics that we've covered are also open to future interpretation and further refinement in future versions of the document. So one of the things that we had discussed quite substantially is whether we could propose a single test media that could be proposed for a specific test method to improve that interlaboratory agreement. Remember our very first slide that was provided by Elijah was that we will have a goal to try and improve that interlaboratory agreement and also improve our read across for other potential materials. And so one of the main topics that we wanted to discuss is, the option of using alternative test media in order to improve that interlaboratory agreement. One of the key components that we discussed was what metric should we use, or in exposure metric should we use. I think everybody is quite familiar with using mass concentration as a metric. And in the guidance document, we state that this is the primary mechanism for our understanding of the particle. But there is lots of other potential alternative exposure metrics. Those include the particle number concentration, the surface area, the surface charge and or surface characterization. So these are all components that the more characterization that you can get done, the better off will be. And as we have advances in more of these techniques, then we'll be able to potentially add them into the guidance document. But we felt that the mass was the easiest to be able to consistently apply, and also for certain labs who don't have all the facilities necessary for extensive characterization, this is the minimal requirement for the test guidelines. You need to be upfront specifically with what types of materials and the interpretations that you have when you're doing your testing, so that you can have a proper interpretation and ensure that you don't have any artifacts falsely giving you results. Next slide, Al. So some of the future revisions that we did discuss was, of course, whether or not and how you deal with settled particles. At least a full day over the couple of meetings were dealt with some of the issues regarding settling, agglomeration and settling, and how to deal with some of the components when more than 20% of your original particle settles out of solution. So it becomes more difficult to test. So the hazard testing for a sample might involve both the suspended particles, the settled particles, and potentially a dissolved fraction, if indeed that's the dissolved fraction does occur. So in some cases, of course, with metals, you might see a significant dissolved fraction at the same time, or you may have dissolution during the testing period itself. And so we described a few different potential approaches which you could be doing testing. One, of course, is to test everything together, which would be in the test media, would allow you to then look at the total toxicity potentially of that particle. And that's the default option for manufactured nanomaterials. And then, but the possibility exists also that we had that perhaps the suspended particles, if they're less than 20%, then you could do a discretionary pretest in which you have the supernatant removed and use that as your exposure metrics, as opposed to testing directly on the material itself. Alternatively, it may call for a secondary testing with a sediment-dwelling organism for particles that have increased percentages or increased amounts of settling over the test period. This will also needs to be informed during long-term tests, chronic toxicity tests, when you might have settling over the longer period, and how you do water changes. And of course, we also had full discussions on the hazard testing of the potential particulate material or dissolution that you would require, and what kind of ultracentrifugation, ultrafiltration techniques are needed in order to ensure that you're testing the undissolved particulate material, or you're testing the dissolved and the potential toxicity, in which then is covered by other guidance documents for metals, for example. An example might be the media manipulations that are needed or not needed when you're working with something like silver, when you would have chloride and or sulfite present. This might change your toxicity potential, and therefore you just need to do the appropriate characterizations to be able to inform yourself properly and have confidence in the interpretation of the test results. Next slide. So I'm going to finish up at this point, but first, before I do, on behalf of both Elijah and Al and myself, as Elijah mentioned, Steve Diamond had a substantial role in the development and the initiation of this document. But I also want to thank a couple other people who over the years have also helped us greatly. That includes Phil Sayer, who is now retired from the US EPA, Doris Volko and Catherine Schwing from Germany. And also thank you very much to Mar Gonzalez, who's kept us in control over the last number of years and has really helped us substantially. So with that, I think we will wrap up. We will remain here for an open question and answer period. You're encouraged to add questions into the Q&A, and or we'll do questions from the in an oral sense from the audience. So thank you very much, and we appreciate your time.
Thank you very much to our three speakers today. I hope you found material presented interesting and useful. We actually have a few questions in the Q&A box. Some of them have been answered already, and we could take a couple more. There is a question on the recommendations on pipetting these stick solutions. For example, how do we account for nanomaterials sticking to pipette tips or glassware? Who wants to take that question?
I'll start off, but I'd also would appreciate suggestions from Greg and Al. I think the glassware part is the easier question because as you're monitoring the exposure concentration, there could be losses from dissolution, from sedimentation, or from sticking to the glassware. So you should get some information about the potential for sticking to the glassware using that approach. In terms of the sticking to the pipette tips, that's a bit trickier. I guess there would be, you'd have to do some preliminary tests to see, to what degree could there be adsorption onto your pipette tips, and maybe compare among different types of pipette versus syringe. What do you think, Alan? Greg, do you have any suggestions for that?
Well, first of all, depending on the type of plastic you use, you're going to have potential for binding. But again, as suggested, I think the easiest way is to do your characterization. If you do it amongst or between a few different types of plastics, you would easily be able to see whether it is something that's consistent or inconsistent between your metrics. So if you're concerned about it, that's the likely way to examine it, to look at the difference between the different types of plastics that exist. But other than that, usually comparatively larger volumes have less inconsistency between volumes. If you're using very small volumes, you may have more inconsistency. So that might be a recommendation.
Good point about the larger volume pipetting. That's really an important point to mention.
And this sounds like it could potentially be a less than stable nanomaterial. So that's kind of one, emphasizes the importance of monitoring throughout the bioassay, especially at the beginning, to confirm how much you actually got into the test media. And now that's contingent on it being a nanomaterial where there are feasible and cost-effective techniques for monitoring that initial concentration. But if it's a particularly sticky nanomaterial to plastics and other materials, that's likely to continue in the test vessel itself. So, for one, maybe pipetting isn't the best way to get the nanomaterial into the media. You may consider things like the direct addition or carrier suspensions with associated controls, so that there's less material lost to the initial surfaces. But this is really where there is no easy button, and case-specific expertise and decisions really are still required. But good question.
Yeah, we do have some suggestions about different types of containers people can consider as well in the guidance document. So if this is something that you're concerned about, you may want to do some preliminary tests with a few different types of containers to see if one has less adsorption than others.
Thank you. We have other questions that are arriving in the Q&A box. There was a request about your opinion about the detection methods for non-soluble nanomaterials like nanoplastic. Do you want to answer?
Sure. I'll start, since we're struggling with this ourselves. I think it's some of the things that you have to be cognizant of plastics, when you're working with nanoplastics, is you have to be aware of the buoyancy potential of your plastics, because often the buoyancy means that your plastics are also either at the surface or are settling. It's something, most plastics have the density less than one, and therefore float. And if you're measuring under the surface, or you're withdrawing your sample from under the surface, your dose metric is going to be incorrect. And so it's a real consideration that you have to consider, that you have to take into account. But in general, the methods for detection of plastics and nanomaterials are similar, but you have to be aware of where and when those plastics are going in terms of density, or also in terms of its ability to be detected by certain certain detection methods. So plastics can often be transparent or translucent enough so that light-based methods don't particularly work well, or alternatively have artifacts. These are some of the things that we're finding as we're working with nanoplastics. I'd be willing to have independent discussions with people because it's certainly something that we need to standardize a bit more specifically for nanoplastics.
Yeah, I'll add a little bit that I think this is an area where that's still very much under development, and it's important to consider if you know what plastic is there, you know, or is there a range of plastics that you're trying to detect? Because if you're trying to do identity, then the different techniques may also be relevant than if you're just trying to quantify the number or the mass or something like that. So it also, there's some nuances about what exactly you're measuring.
And I will add that we had some discussion about micro and nanoplastics. The focus of this guidance document, of course, was on manufactured or intentionally designed nanomaterials, not necessarily things like microplastics, which are more incidental. I hope we're not purposely making those. And there isn't
applications that are intentional microplastics.
at least not in the ocean, hopefully. Yeah, so there is minor discussion of that. I know we've included in some white papers and other documents that some of the methods may be applicable, but there isn't much discussion in the guidance document itself, and the characterization of those materials is particularly challenging, at least compared to things like nanometals. It's more like the carbon nanomaterials.
Thank you. There is another question that is relevant to the topic discussed today. For classical substances, we generally express the results as ECX, effect concentration 50, or NOEC, or based on dissolved concentrations. How to proceed for nanomaterials? Should we express the results, ECX or NOEC, on the basis of the total concentration to take into account the dissolved, suspended, settled fractions? If yes, should this total concentration be used for a classification purpose? So there are multiple questions in there, but I guess you have elements of answers to share with the attendees today. Who wants to start?
Yeah, I'll try to start while the other two think about the question. It's certainly a stacked and very good question in terms of its content. So this again is where the guidance document goes into a lot more detail than we're able to in the presentation. But the default, as you well know, is mass concentration. For many materials, that has been the default for a long time. For nanomaterials, I think there's agreement that there's probably more robust methods for characterizing the exposure. But in this guidance document also, that's the default consideration, with the assumption that determining things like dissolution kinetics may be a bit easier than routinely characterizing particle number or surface area metrics. That's certainly encouraged, but the default is mass concentration. And you know that there could be inaccuracies or lack of reproducibility for materials that are behaving dynamically, but it's usually the more easy to measure metric for many materials such as metals. But the guidance document very much encourages getting additional information that could facilitate more descriptive toxicological endpoints, such as a surface area based equivalent to EC50. Of course, you couldn't use the C anymore. And then I invite Elijah and Greg to expand upon what I said.
Just to maybe just add very briefly, in those jurisdictions where you are allowed to talk to your regulator, I would potentially ask for advice or guidance as what they will be accepting. But regardless, we all know that mass concentration is not the ideal metric, but it is one that you have to report. And then after that, if it's going to help inform your the potential use or the end use and the end receptors that are necessary, then there's guidance on the potential for testing, perhaps in the sediment and in the sedimented particles, and in a sediment testing, and then a pelagic-based testing for suspended particles, and also for separating out the independent effects of the dissolved material from the nanomaterial. If you can do some of those tests, then you're allowed, that increases your confidence in your information as to what that mass-specific LC50 NOAC or LOAC actually means.
And the other part of the question that we so far haven't touched is, I guess there were three major debates. There were a lot of debates, but three come to mind. One was on that 20% guideline and whether that should be applicable to nanomaterials. Another one was, having discussions with regulators and whether that was allowable regionally. Some places it is. We take advantage of that here in the US in our dredge material program. But the last one was whether the settled fraction or mass of the nanomaterial should be included in the total dosimetry or exposure concentrations. And the pros and cons of doing that, and I briefly mentioned that, and it's briefly mentioned at the bottom of one of the flow charts, is the default is considering the total mass of the nanomaterial added to the test system. But there were experts that also believe that the focus should be on just the suspended fraction. And a lot of the toxicity of the metals could be potentially controlled by ions dissolving off the metals and the rate that they're dissolving. So really, all three of those options are available, and that's where you can gain more information on what the animals are actually responding to by, for example, testing a supernatant, not including the nanomaterial that's settled, as well as a more rigorously centrifuged supernatant, which may include the dissolved fraction for metals. So that was very much a topic that was up for debate and we didn't have full consensus on, which is why the document doesn't provide definitive guidance on exactly what to do. But some felt that, sort of the nature of nanomaterials settling in interactions with the settled particles in the organism are a pertinent consideration. But I think it was agreed that that adds to the variability of the results, kind of looking at physical versus the more traditional chemical toxicity.
Thank you.
Okay, I had one piece before we move on to the next
one.
One thing I want to emphasize, in addition to what Al and Greg said, is just the importance of quantifying the dose. Regardless of which one you choose, it's really important to have quantitative information about what exactly you're measuring.
Thank you, Elijah, for this additional thought. Another question maybe we could take today is a participant expressing concerns about the artificial definition of nanomaterials, one dimension below 100 nanometers, and its consequences regarding aquatic toxicity testing. According to OECD guidance document 23 on the difficult to test substances, testing is invalid using test suspensions containing non-dissolved test material to avoid the physical effects, as you know, on the test organisms. While for nanomaterials, one is to include particles into the test. There should be a practical, viable testing strategy for non-intended nanomaterials being at the boundary between bulk and nano. Any reflection? That's, I guess, not exactly a question, but more of a comment, inviting considerations from any of you if you want.
Yeah, I guess I'll make 2 points. One is that we very intentionally in the scope of this document didn't want to get into the nanomaterial definition question, and different jurisdictions may have different definitions of what is a manufactured nanomaterial. And so we just didn't touch that topic. The second topic is like how to handle this issue with like the non-dissolved fraction. And I think this is, this gets into the physical effects, and kind of the last thing we talked about with the different fractions of settled or just a suspended and so forth. I think this would, if you're in a jurisdiction where you're capable of talking with the regulator, this would be a good discussion to have if you're wondering, do I go down the guide, the route of guidance document 23 or 317? You may be able to get some guidance there.
Thank you, Elijah. I don't know if the other two speakers want to add anything. If not,
no.
Do you want, Greg? Or?
No, thank you. I think Elijah covered it well.
Okay, good. There's maybe one or two last questions we could take today. One of the attendees today is interested to know, what do you see as the main nano-specific challenges for sediment toxicity testing specifically? Characterization of the nanomaterial in the complex sediment matrix, and or anything else?
Okay, well, I'll start. That's a great question, and something that we didn't specifically cover in the presentation. A lot of the focus was on water column exposures, and as I'm sure the person who asked the question knows, there's less available published information in the literature on sediment nanomaterial type of exposures. So what the guidance document does do, and there's approximately 2 pages dedicated to this, is that it focuses on the way to spike the sediment. And there's been a number of things that have been attempted in the literature, such as spiking the water column and allowing the nanomaterial to settle to the sediment, which is sort of thought to be a natural type approach of how nanomaterials may settle in the environment. And it would be more of an epibenthic or superficial exposure. Perhaps the more customary spiking method is to directly add the nanomaterial, and there's several different methods discussed on that, to the sediment and homogenize it. So what the guidance document does do is discuss the homogenization methods to get the manufactured nanomaterial well mixed into the sediment, so you don't have hot spots, and that is like a cluster of nanomaterials in one portion of the sediment, and then another portion of the sediment is fairly devoid or clean from the nanomaterials. You want a consistent exposure. But of course, it's complex, and the most important consideration is being able to actually characterize the nanomaterial and its state in the sediment. And that's what's so difficult. The easiest thing to do is for metals, do the traditional extraction for total metals. And of course, that tells you nothing of the pore water concentration, the particulate or solid phase fraction of the nanomaterial. And that was basically a knowledge gap that was acknowledged in the guidance document, that there isn't enough published in the scientific literature on sediment assessments, and why we focus so much on very efficient and thorough homogenization protocols and characterizing the material before it goes into the sediment. Because for a carbon nanotube in a sediment, characterizing it after it's actually added to the sediment is more complex. And I appreciate a bailout from the other 2 presenters.
I don't think you need a bailout. I think that was pretty thorough. So I'll second what you said.
Yeah, there are issues, of course. Once you get into the characterization, once you get into the sediment, is quite difficult, especially in particular cases like, as Al mentioned, carbon nanotubes. There aren't really great detection methods in that particular matrix, but there, again, the complexity of the nanomaterials is starting to come out here. The same arguments that we had over and over again in meetings are coming out, and we're aware of these difficulties, and hopefully with future research, development of techniques, we'll be able to answer them a bit better. This is the first attempt at trying to get the guidance out for testing. But thank you for the question.
Okay, we are going to stop with the questions and answers live. So if the speakers want to have a look at the remaining questions in the Q&A box and provide responses in writing, while I wrap up and conclude on this webinar. Thank you very much to our speakers today for sharing your knowledge with many participants, more than 150 on the webinar today. This is great. So now we are going to conclude for today. So on the slide shared on the screen, you can see where to find the video recordings that will be made available very shortly in English and Spanish on our website. And also, if you want further information, you are invited to look at our website. If you want to receive the newsletter from the Chemical Safety Program at the OECD, you can sign up to our newsletter and follow us on social media and visit our YouTube channel where we have a lot of videos of webinars and beyond nanomaterials as well. Also, an announcement, we will have another webinar next month and you are invited to register. This will be a webinar on assessing the dispersion, stability and dissolution rate of nanomaterials in the environment. So this will be a related topic to what was discussed today. And you have the link for registering to that next webinar. So with that, I would like to thank you very much today.